This is the G2 phase, which is just the checkpoint on the cell cycle. And what happens here is just looking for errors and growth on the DNA strands that are duplicated from the origin. So, for example, if an error were to occur on this checkpoint, this regulation monster would come and just say no. That would be the process. Like, as example, the two DNA strands that come from the S phase come along, and you have one of the DNA strands have an error. In that case, that error would be obviously dealt with, and then that DNA strand that is fine, it'll just continue onto growth and end in mitosis.
G1 phase stands for gap 1. Interphase begins with this phase. During the G1 phase, the cell make a variety of proteins that are needed later for DNA replication in S phase. Not all cells continue to replicate, and in that case, they would the cell will enter G0 phase from G1 and only to re-enter G1 phase to begin dividing again under specific condition. So the cell will increase its size and producing RNA and synthesize protein. It will grow larger and getting larger and larger. And after that, it will go to checkpoint, the G1 checkpoint, the safeguard that ensure the protein synthesis occurred properly and the cell DNA is intact and ready for future stage.
The last phase of cell cycle, which is mitosis, and now we're gonna start the prophase, which is the first phase of mitosis. At this phase, the chromosome condense into the compact structure that you can see right here. After that, the centrioles start moving with the microtubules, like you can see some of them right here. It starts to moving over on the top, and same at the bottom. After that, the nuclear envelope starts breaking down, which is stepping up to the metaphase, and these microtubules that you can see it have more of them. And it start connecting to the centromer of the chromosome. Like you can see like that on both sides, and each of these, like you can see, is a sister chromatic. After that, the next phase is metaphase. During this phase, the microtubule have a function of moving and pulling the sister chromatic up to make sure it line up to each other, which you can see like this. It makes sure it line up so it can ready for the next phase. Start connecting, still connecting into the centromers. At this phase, this black thing right here is called kinetochores.
Kinetochores.
It's called kinetochores, which is the part that's sticky to the microtubule, and it's helping to sending signal with using the motor protein. The motor protein gonna moving from the kinetochores, moving from that into the centrioles to start signaling when is the moment to get these apart. Now we start to moving to the next phase, which is anaphase. At this phase, the force of the two meiotic spindle chromosome toward opposite poles of the cell is have been pulling these apart like that, and it break the link joining between each pair of sister chromatic and make sure the daughter cell receive the same amount of chromosome. So stop moving like that, and it stop pulling apart, and it stop moving on each side. And after that, it start condensing, which is moving to telophase. The sister chromatic start to condensing with the centrioles. At this time, the microtubule is gone. Let me take this out. Microtubule is gone, and it starts to forming the nuclear envelope starts to forming again around the set of chromosome. And like I said before, on each of these rounds, it makes sure that it have enough of chromosome on each individual one. After this phase, mitosis is complete. Now we're moving to the cytokinesis, which is the last part that the cell start to dividing. Cell start to dividing. And at this phase, the cell membrane break apart. No, it's not break apart, it's actually pulling apart and start forming another cell right here that you can see, and everything is inside there. And both of these start moving out there and out here. And like you can see, the bubble one right here, this one is the actin filament, which is kind of like the cytoskeleton, which is helping keeping the shape of the cell, and keeping the structure so it can moving frequently.